Hi guys, welcome to my second inheritance video. Following on from my introduction to inheritance, this is application of genetic inheritance. So we've looked at the basics of monohybrid and dihybrid inheritance and how to lay it all out. And now there's a few extra sort of exceptions to the rule and extra things we need to know about. Okay, so the first thing I want to talk to you about is codominance. So far we've known that in allele pairs there's always a dominant and a recessive one. Not true. Sometimes they can be codominant, like the colour in petals. So in this case, if you have white petals and red petals inherited, you can get, if it's a heterozygous allele, pink petals being made. So the colours merge because they're codominant, so they're both expressed, both alleles are expressed in the phenotype. You can spot codominance because they're usually written sort of in this way, with this sort of power of letters. So for white, you've got colour W, so CW, and colour red, CR. Okay, and then pink would be CRCW. Make sense? Right, so the next thing I want to talk to you about is multiple alleles. So far we've only looked at when there's two alleles, you know, big B, little b, but sometimes there can be more than that, like blood group, which has 3, which has these ones. They're always written this way because some of them are codominant. So IA and IB are codominant, and IO is recessive to both of them. So here's all the different combinations. Can you see how with A and B, if they're heterozygous, IAO or IBO, the IO is recessive and isn't expressed, and only the dominant A and B is expressed. But when they're together, IA, IB, you get AB, and then you only get O if you've got both the recessive alleles. So it's a bit rarer. Right, so try and pause this video and have a go at crossing a heterozygous A group person with a heterozygous B group person and see what possible offspring there are. Okay, so pause the video now. Right, here are the answers. So we should be getting 25% of each different type of blood group. So 25% AB, 25% A, 25% B, and 25% O. Okay. The next thing I want to talk to you about is sex linkage and other types of linkage. So sex linkage is when you get genes on the X chromosome, the sex chromosomes. You don't really get many genes on the Y chromosome because it's pathetically tiny and there just aren't enough room for many genes on it. You might have a question with something on the Y chromosome, but to be fair, they're mostly going to be on the X chromosome. So here's an example for you. Haemophilia is a sex-linked trait which is carried on the X chromosome. X to the power of H means you're normal, and X little h means you've got haemophilia. So as we know, females are XX and males are XY. So we've got a carrier female here, which means she's got a big H, little h on her X chromosomes. And we've got a normal male who has inherited the normal X, big H chromosome. And as you can see, Y doesn't have this allele on it. Right, with males and female crosses, you will always get 50% male and 50% female, but that's not what we're looking at. We're looking at whether they've inherited haemophilia or not. So this female here is totally normal, 2 sets of normal female. This female has got 1 normal allele and 1 haemophilic allele, so she's a carrier again, like her mother. And then we've got the 2 males, which only inherit 1 of each X chromosome, so they either have it or don't. This is called being hemizygous, when you only inherit 1 form of the allele. So males are hemizygous in this case, so you either get a normal male, like this one, or a male with haemophilia. So sorry boys, but you're more likely to inherit these diseases. You can also have autosomal linkage, which means all of the other chromosomes, so anything that's not a sex chromosome, so all the other 22 pairs, if you have the same allele on the same chromosome, it can affect the overall outcome of phenotypic ratios. Okay, and you've got a list of those in both of your books, so you just double check you know what linkage means for the outcome of some inherited phenotypes. Okay. Next thing I want to talk about is epistasis, which is when genes interact with other genes in the expression of lots of other genes. So it can be found in enzyme pathways. You can get one gene affecting the expression of the next protein and the next protein and the next protein. So it's kind of like another level of control, I suppose. You can get dominant epistasis or recessive epistasis. Which one should we look at first? Here's dominant epistasis, which is colouring in squash, like, the vegetables. It's controlled by two genes, a white allele and a coloured allele. Okay, so first of all, if you inherit the coloured allele, which is little w, you can then go on to inherit this yellow or green allele, which will eventually give you a yellow or a green squash. But because this is dominant epistasis, if you have a dominant coloured allele, so big W, little w, or big W, it blocks that coloured allele, recessive coloured allele, from working, and you get just a white squash. This also works the other way around with recessive epistasis. This is another example of colour, which is just a bit more obvious to look at, I suppose. So this is flower petal colour. Gene 1 codes for a yellow pigment, and gene 2 codes for an enzyme that turns the pigment orange. Right, so if you have, let's go down here, colourless molecule, so if you inherit the big Y, or big Y, little y, then the yellow pigment can work and either be yellow or orange. But if you inherit little y, the yellow pigment and orange pigment cannot be expressed. So gene 1 is epistatic to gene 2, because if the gene is little y, gene 2 will never be expressed. So you need those dominant alleles, those dominant big Ys in there somewhere, otherwise you never get onto the this R allele to express yellow and orange. There's a lot more detail. I've got these examples from the guidebook, the OCR guidebook, which is done by CGP, and those examples are quite clear, especially when they're colour examples. They're a bit easier to understand, but you might get one in your test that's nothing to do with colours, but it's still look out for epistasis. Okay. And finally, the statistical test. So all of this inheritance stuff has been about predicting what the outcome is going to be in the offspring. But because we know this is only a prediction, we might get 50% blue and 50% red. But in real life, you could have 100 offspring and you still get them all red. It's just a probability. Yeah. So the chi-squared looks at the difference between what you expect, E, against what actually happens, the observed ratios, O. Okay. So I don't know, perhaps you've got a field of crops and you were expecting to get about 50% one allele and 50% the other, but you got 75% and 25% instead. And you want to do a little statistical test to see if it's significantly different from what we expected. So here's the formula, but it's much easier to use a table like this. I'll talk you through it now. So here's my example, the stem of this question. In Drosophila melanogaster fruit flies, big N is normal wings and little n is vestigial wings. A big N is crossed with little n, and in the F2 generations, that's the second generation, there is a 75% chance of normal wings and 25% chance of vestigial wings, ratio 3:1. Okay, so normal wings, vestigial wings, ratio 3:1. And 160 offspring were produced, so you'd expect 120 normal wings and 40 vestigial wings out of 160. But what was observed was 111 normal wings and 49 vestigial wings. So the first thing we do is find the difference. So we've got -9 for that one and 9 for that one. And then you square them, so that brings them back into positive numbers. And then you divide that by E. So that would be, in this case, 81 divided by 120. And for this one, it would be 81 divided by 40. And then you add them together, and that is your chi-squared value. So what do we do with that? Well, first of all, we need to figure out our degrees of freedom, which is always n minus 1. And in this case, n refers to the categories we've got. So we've got two categories, so 2 minus 1 is 1. So we've got 1 degrees of freedom and a 5% probability value. Now, I don't have a lookup table here, but if you go online and type in chi-squared lookup tables, you can have a look at what this looks like. So I looked up the critical value in these tables, and the critical value was 3.84. And here's what that means. If the chi-squared value is more than or equal to the critical value, you reject the null hypothesis, and if the chi-squared value is less than the critical value, you accept the null hypothesis. So in this case, the null hypothesis would have been there will be no significant difference between observed and expected values. So our chi-squared value is 2.71, which is less than the critical value of 3.84, so we accept the null hypothesis. So this is true. There is no significant difference between observed and expected values. We know there is a slight difference of 9 for each one, but because we've got quite a big overall number, 160 offspring, it's not a statistically significant difference. Right guys, I am taking requests for things to cover in your A-level biology course, so if you've got something you really want me to cover, let me know and I'll make a video about it.